many people over the age of 55, 60 in our culture are hyperinsulinemic. They're not yet diabetic, but they're sort of on that path, generally speaking, and they have elevated levels of insulin, but the glucose is not high enough yet to meet standards for treatment as diabetics. But I think damage is still being done in that phase, and that phase can be very long. It can be 5 years, 10 years, or longer.
Thank you, Dr. Krikorian, for being here. This is a really exciting interview. I'm really excited to talk to you. You've done such exciting and incredible work. So just before we begin, thank you for taking the time to talk to me.
Oh, you're welcome. I'm really pleased to do this.
Of course. Before we get into the science, I would love to just hear some of your backstory. How did you decide to really focus on this in your research? That is the way that diet and lifestyle really mediate cognitive function and risk for neurodegenerative disease.
Well, it's something that it's an interest that developed over a period of time for me. I formerly had done some research in instrument development. I'm a neuropsychologist by training, which means I'm interested in brain-behavior relationships, in particular cognition. And that could be anything from developmental learning problems or attentional problems like ADD to cognitive problems that are associated with brain injuries or psychiatric problems, and of course, cognitive changes with aging. And previously, I'd done some research looking at developing some new instruments, and we did that. We developed several instruments, new measures of attention and executive ability and long-term memory, and some of those we actually use in our research now. And a long time ago, I got involved in a little bit of HIV research. A psychiatrist and I were the only people in our department at the University of Cincinnati Medical Center who were involved in that initially, at least, and I published some papers in that area.
Wow, your research really runs the gamut, or at least has in the past.
I've just followed my interests and opportunities as they came along, and I've done that my whole career, my whole intellectual life. I mean, I started out being interested in astronomy and cosmogony, and I went to undergraduate school initially and majored in astronomy, and then I switched to philosophy and got my undergraduate degree in philosophy, and then I went to graduate school in philosophy for a couple of years, but decided I wanted to move into psychology. Got a terminal master's and in philosophy, and then I thought I was going to be a psychoanalyst, actually. But in my PhD program, I wound up doing 2 research studies, neuropsychological research studies, and then I got some clinical training in neuropsychology, and that kind of changed my career right toward the end. And I got into neuropsychology in my first actual job. And then, as I said, I just followed my interests from there. So I have a clinic, and we see patients from age 7 through old age.
Wow.
And I would guess about 8 years ago, maybe 10 years ago, we started to see more people who are middle-aged and older with age-related memory changes. And the typical patient was sort of dysthymic, had a little bit of depression and increased anxiety, and starting to have some cognitive changes. And I noticed some things about them in terms of, just sort of simple observations about the other health conditions they had, like typically hypertension, sometimes type 2 diabetes. They seemed to be overweight as a group. And it just occurred to me that this was going to become a major issue in the population as a whole. I remember actually I have this memory of one day sitting in my office in mid-afternoon looking out the window and thinking about this. And I guess that was a moment in which I sort of found a new interest area and started to pursue it. And since then, I've done a lot of non-systematic, but just my own reading about lifestyle factors. And typically, people think of stress, negative stress, and physical activity level, and nutrition or diet as the big three lifestyle components. And I certainly would agree with that. And I've really focused on nutrition in my research. I think it's the most potent factor in influencing our health and longevity and risk for late life diseases, of course.
Wow. I mean, based on everything I know, I would completely agree with that. I'm just curious. So many researchers that I know that focus on prevention do so because they have a family history of Alzheimer's disease or other neurodegenerative disease. Did you have anything like that in your family tree?
Actually, no. My parents lived a long time, and my mother is actually alive, still alive, and relatively healthy. She's suffered cognitive decline in the last few years, and it's related to some medical conditions that have caused encephalopathy. But otherwise, dementia is not a big factor in my family, which I guess I'm lucky. I mean, I know it affects many people, actually most people, I think.
Yeah, people even without known genetic risk factors. I mean,
right.
And I would, not to make the conversation about myself, but I'm one of those people. I didn't have a family history of neurodegenerative disease, and my mom, it's not a secret that she has a kind of cognitive decline that she's experienced. So yeah, I can definitely one thing that I like to underscore is that these ideas that we're talking about are applicable to the entire population. I mean, really, there's nobody that's not ultimately at risk. So one of the studies, before we became friends, I was actually aware of your work, and then it was just, so amazing to have actually, built a rapport with you. You put out one of the sort of seminal papers that looked at the impact of dietary ketosis on cognitive performance in people with mild cognitive impairment. So just, to recap for listeners that don't know what dietary ketosis is, it's what happens when you are either in a fasted state or you deprive the body of carbohydrates, and your brain then taps into fat stores for fuel. And, fatty acids are liberated. They become converted to this alternate fuel source called ketones. The brain can then use it. So why did you decide to study people with mild cognitive impairment? Why did you pick that group? And then why did you decide to look at ketones and how they might affect their cognitive function?
Well, we did that research a few years, actually over a period of probably 5 years. We had almost no funding. Wow. But we sort of kept at it and kind of utilized resources that we had from other funded projects, but took a long time to complete it. But in any case, I became interested in mild cognitive impairment when, because it was a quote pre-dementia condition. It was not Alzheimer's disease, or it was not dementia, and so again, it for probably not a great reason, or not a systematic or empirical reason, but my sense was, I'm not a physician. I'm a PhD, and so I don't have access to medications as treatment tools. And so, as I mentioned earlier, I was focused on nutrition, stress control, and exercise. We actually did a small exercise study a long time ago. But in any case, with those tools, I thought again that diet or nutrition was the most potent way to approach this, and I really felt strongly that prevention was much preferred relative to cure. So it's and it's easier, and I still feel that way. It's I don't know if we can cure Alzheimer's disease. There are some people think it's once you get to that stage, the pathological factors are almost self-reinforcing, and effects become causes and so forth, and there's a sort of a synergy in a negative way about the pathology. So it's very hard to intervene, and that's been demonstrated because so many promising compounds, pharmaceutical compounds, have really just not worked. And it's interesting we're talking about this because just this week we actually do clinical trials in my program. We do my research, but also some clinical trials, and 2 of our studies have been terminated because the agents don't look promising. Just this week, we got notices on one day. I think it was Wednesday from one company, and then the next day from the other company. So it's kind of timely to talk about it. But in any case, I really wanted to focus on prevention, and I had already become interested in berry fruits and some of the basic science in that area. But I knew a little bit, I guess not enough to be dangerous in terms of ketone metabolism, and it was just intriguing to me. It still is. I'm really still very enthusiastic and excited about this intervention, and I just wanted to study it. So we did, and that's kind of the way I've stumbled into things. And I know a lot more about it now than I did back then. But we designed a study. We designed a controlled trial, again, a prevention trial. So it was MCI participants, and it was a very small study, as I mentioned, and it took us a while to complete it. But we randomly assigned people with mild cognitive impairment to either a higher carbohydrate intervention, which is basically about 50% or more of their calories from carbohydrates, which is somewhat typical of the standard Western diet. But we asked those people in the higher carbohydrate group to try to clean things up, try to eat more vegetables and fruits than processed grains and sweets. We asked them to use olive oil for fat. We wanted it to be perceived as a healthy diet.
A healthy high carb diet.
Healthier, yes.
Yeah,
a healthier
healthier
diet, yes. And in the low carb group, I decided to use the first phase of the Atkins approach, which is a ketogenic phase. I think he refers to it as induction. So not, counseling people to increase their fat to get adequate calories, but really focusing on carbohydrate restriction. And what we prescribed is 20 grams a day as the limit on carbohydrates, which is pretty drastic, actually. So no fruit, we disallowed all grain-based foods, and of course sweets, no calories from liquids. And we asked them to consume vegetables in modest portions 1 or 2 times a day, and then to eat freely protein and fat, and emphasize that increasing fat would be a good way to get calories. And it was just a 6-week intervention. Initially, actually, we did a 3-week sort of pre-pilot just to see if people could manage it. These people are, on average 72 years old, and we weren't sure that they could carry out the intervention. But it looked good in 3 weeks, so we did the full 6 weeks, and it was kind of extraordinary. When I finally got to look at the data years down the road, we got this nice effect for improvement in memory in just 6 weeks on a really relatively challenging memory task. And we also found that they lost a bunch of weight. I think it was approximately 15 or 16 pounds on average. They lost a couple of inches on waist circumference. More importantly, fasting glucose and fasting insulin came down in the low carb group. And that was, among the most gratifying results, and of course expected. I looked at associations of weight loss, change in calorie consumption, and a few other things with their memory performance, and none of those factors were associated with the change in memory. So it wasn't reducing calories, it wasn't the weight loss. But within the low carb group, within the ketogenic group, there was a a trend for a moderate correlation in the 40s, zero point 0.4, I should say, with insulin, and it was an inverse relationship. So as insulin came down, cognition improved. And that was one of the things I was interested in, because I thought that if we could correct the hyperinsulinemia in these people, many people over the age of 55, 60 in our culture are hyperinsulinemic. They're not yet diabetic, but they're sort of on that path, generally speaking, and they have elevated levels of insulin, but the glucose is not high enough yet to meet standards for treatment as diabetics. But I think damage is still being done in that phase, and that phase can be very long. It can be 5 years, 10 years, or longer, I think. But in any case, we lowered insulin into the reference range, and it was just at the upper limit of the reference range as they entered the study. And glucose came down to the mid-80s, which was great.
Wow.
A lot of these people were, around 95, 99, 102. They were borderline diabetic, as I was saying. So those were really great results. But I thought part of it was the correction of hyperinsulinemia, but I thought that there was another component which might have been an energetic component. That is, we were improving mitochondrial energy production in the brain. And I did publish another work, which you may not have found, because I think it's in a very small journal that's not, well, it was actually started by a few friends of mine, so I published these data in there. But we just had, we did an extension study. We did 5 more people, all of whom went into the low carbohydrate arm, the ketogenic arm, and we did pre and post memory testing, but we also did brain imaging. And the procedure that from which we published the data was proton MR spectroscopy, and that's a way to look at certain neurochemicals in the brain. And when we did pre and post spectroscopy with these participants, and so they showed many of exactly the same metabolic changes that we saw in the first larger study, and also showed improvements in cognition that were similar. But in addition, they showed a significant increase in a neurochemical called myoinositol, and we can talk about that in a moment.
Wow.
As well as trends for improvements in N-acetyl-aspartate and creatine and phosphocreatine. And we don't have to get into the details of that if you don't want to, but they are sort of provocative. And this is just, again, a very, very small study, uncontrolled, but I think the data is very interesting and needs to be pursued. And we are, in some sense, today with the new study.
Creatine is something that I've definitely done a dive into. Its ability to improve cognitive function in vegetarians who don't intake a lot of dietary creatine. It's something that's naturally found in the brain. It's really important for brain energy metabolism.
Yes.
And it tends to be lower in ApoE4 carriers of the genotype that puts a person at higher genetic risk for Alzheimer's disease.
Right.
But I hadn't heard of the other chemicals that you mentioned, so I would love if you could sort of touch on those.
Well, sure. And this is, out of my area of specialty, certainly, but I'll just say a few things about it. Myoinositol, we did get a significant statistical effect for myoinositol, and that's a chemical that when I read about it, it seems like no one knows really what it does. I think it does many things, actually, but it seems to reside in the cell walls. And it's actually been used as a marker of neurodegeneration when it's looked at in longitudinal scans over time. And as people with MCI or Alzheimer's disease, for example, progress, you see reductions in their cognition, you see reductions in N-acetyl-aspartate, something else we measured, so that tends to get lower, and myoinositol tends to increase, and the idea being that as the cells are being destroyed or succumbing to the neuropathology, this myoinositol is being liberated, so to speak, and is being imaged because of that. But we had sort of the opposite context. We had increased N-acetyl-aspartate, and we had improved cognition, and we have an increase in myoinositol. So it doesn't seem to be in our study then a marker of neurodegeneration. Um, I I can't prove this. We certainly need to look at this again. But my suspicion is that it's an upregulation in what's called autophagy.
Hmm.
Autophagy is you may have heard the term. It's something that's upregulated with ketone metabolism, and it's a normal housekeeping function where um, cells that are dysfunctional or dead or for other reason impaired are broken down. In a sense, the chemical constituents are liberated for use in other new cells.
Hmm.
So it's an interesting process. I think it also has a role in cancer prevention. Um, and so my guess is that that strong effect we got with just 5 people was an upregulation in autophagy associated with the ketone metabolism, which would be very interesting. And again, I would like to have a chance to look at that further. We got a trend for improvement in N-acetyl aspartate or increase in N-acetyl aspartate, which is a relatively well-established marker of neuronal integrity. So that's, again, encouraging. And then this other marker, creatine plus phosphocreatine, it's a combination. And as you were saying, those neurochemicals are very important in terms of, or those chemicals are very important in terms of energy metabolism and energy production. And phosphocreatine is sort of a storage buffer of energy that's created by the mitochondria. Again, that was a trend, but only 5 subjects, so it's provocative, and we need to look at that further. So, very small, uncontrolled study, but a lot of interesting data came out of it that supports this notion of potentially improved energy production with the ketogenic diet, and then perhaps improved defenses or an upregulation of beneficial processes in the brain as well.
I think it's really interesting that you mentioned that you focused not so much on having your participants eat more fat, but instead reduce carbohydrates. I think there's a lot of, the ketogenic diet has become a bit trendy as of late, and people tend to think that they need to eat lots of fat to achieve higher levels of ketones. But you're saying that it's not really so much, ketone synthesis in the body is not so much a function of lots of fat consumption. It's really mostly about reducing carbohydrate consumption.
I think so. Yeah, I think it's. I mean, the modulator of ketone production, and really the fail-safe on ketone production, is insulin. So the reason to reduce carbohydrates is because carbohydrate is the principal determinant of insulin secretion. So you're bringing carbs way down, and then you're also bringing insulin down. And in that context of low carbohydrate and low insulin, the liver will produce ketone bodies. And that's the principle we followed. Now, we did, a 6-week study, and more recently, our current studies are 8 weeks. So we've extended it a couple of weeks. And in our studies, those short duration studies, what people are actually doing, we do measure their intake with diet records, diet diaries, and what they're doing is essentially cutting out the carbohydrate consumption. They're not really significantly changing their protein intake or their fat intake. So they get a pretty large reduction in calories per day, and that goes on for 6 or 8 weeks. But my sense is if they continued on this approach for a much longer period of time, their weight shedding would start to dissipate, and probably they would start consuming more calories per day. I'm almost certain of that. And if you want to stay at that stage in a ketogenic diet, then you need to increase fat, I would say. Because a ketogenic diet, it's not a high protein diet. That's a misconception some people have. It's a moderate protein diet. And actually, if you eat too much protein, you'll sabotage the ketosis, because there'll be a glucose generation, protein stimulates an insulin response that's approximately 50% of the insulin response that carbohydrates stimulate. So
Interesting.
um, you don't need to starve yourself of protein, but you can't go too high either. So the only thing left to modulate then is fat, and that's also a natural for a ketogenic nutritional approach.
Right. So you've mentioned already a few of the, you've talked about some of the potential mechanisms that were at play in terms of the observation that you saw on your subject's cognitive function, the reduction in insulin. But what is it about ketones specifically that might help the brain of a person with cognitive impairment?
There were actually a few things that seem to be apparent in basic science studies and animal studies that, to my knowledge, haven't been looked at very much at all in humans. I think this is a field that really demands more investigation from a mechanistic level and also a clinical trial level. But in any case, one of the things, if we're talking about neurodegeneration and late life dementia, one of the things that has been demonstrated in animal studies is that about 8 weeks of ketosis in mice, I believe it was associated with reductions of beta amyloid and I think also hyperphosphorylated tau.
Wow.
So those are, as you know, cardinal pathological features of Alzheimer's disease. So you're seeing a reduction of the pathology of a prominent neurodegenerative disease. So that's quite important. And there are ways to look at that in humans, of course, that aren't terribly invasive, but are somewhat. And ketone metabolism is also, I don't know this literature quite as well, but it's associated with a reduction of inflammation.
I think it's been shown recently also to have an epigenetic, these ketone bodies have been shown to have an epigenetic effect and help, I think in animals maybe, but to help upregulate production of BDNF, which is brain-derived neurotrophic factor. It's a really important protein for, neuroplasticity and things like that. But then there's also, I'm pretty well versed in the idea that there's this sort of, at least in Alzheimer's disease, this reduced ability of the brain to create energy out of glucose, which is sugar, essentially, which for most people, especially those on the standard American high carbohydrate diet, it's pretty much what the brain is running on full time, is sugar, essentially. And ketones, I think, provide an alternate fuel substrate, allowing the brain to create ATP, allowing essentially a diabetic brain to create ATP with an alternate to glucose.
Yes, and that's I think that's a little bit controversial, but I tend to agree with what you said. Stephen Cunnane, who's a friend and colleague at Sherbrooke University, somewhat relatively close to Montreal, has done a lot of work. I mean, much of his career recently has been devoted to looking at ketone metabolism in the context of Alzheimer's disease. And he's shown in publications in the last few years that the Alzheimer's brain, I mean, just to back up for a moment, the non-Alzheimer's older brain has a glucose metabolism deficit of about 8%. From a whole brain point of view. But the Alzheimer's brain has a deficit that's about triple that. So it's close to 25% of glucose hypometabolism. That is a deficiency in the ability to utilize glucose. And he's looking at mild to moderate Alzheimer's patients. So presumably that would be even greater in the later stages. And yet older adults, healthy older adults, and older adults with Alzheimer's disease are not deficient in their ability to utilize ketone bodies. So that's a crucially important fact with respect to ketone metabolism as a treatment for late life brain disorders, so that in particular Alzheimer's disease. So even if you have Alzheimer's disease, your brain is still able to use this alternate to glucose for energy generation. And the other thing about ketone metabolism is if you do it with a nutritional approach or a dietary approach in the way we do, you're actually also increasing the brain's ability to utilize glucose. You're kind of tuning that process up as well. The glucose scarcity seems, and I think also the peripheral, the level of insulin in the periphery, the reduction of that is helpful too. I think part of, I'm not sure if it's type 3 diabetes in the brain, but I think what may be going on, at least in part, is when you have peripheral hyperinsulinemia, much of the insulin in the brain has to come from the periphery, has to be transported into the brain, and it's a receptor-mediated transport system. So those insulin receptors in the transport system can become resistant to the effect of insulin in the same way muscle receptors in muscle tissue or other tissue in the body can. And so essentially what you can develop is a condition of peripheral hyperinsulinemia and central hypoinsulinemia. And insulin really does a lot of good things in the brain, and it at normal physiological levels. And in addition to getting glucose into the tissue, it helps with new memory formation and new learning. Also, level of insulin is very closely tied to an enzyme called Insulin Degrading Enzyme. And that enzyme, one of the functions of that enzyme is to help get beta amyloid out of the brain into the peripheral circulation so it can be eliminated from the body. So it's really, I think, a matter of the brain, and this is not demonstrated certainly to a full extent, but it seems that it may be a matter of not enough insulin in the brain to help glucose get into the tissue, but also to do some other of these housekeeping functions that are very important in maintaining good brain function.
So just taking, a step back and maybe because as you've mentioned, prevention is so important, and, it's definitely a passion of mine. I know that some research has shown that, some of these metabolic deficits that are evident in the aged brain are pretty similar in people that are very young with certain genes.
Yes.
That predispose them for increased risk for Alzheimer's disease. So what do you think the implications are of your research and, the application of a ketogenic diet, perhaps, or diets that allow for ketone generation for young people?
I think, well, you're asking me the perfect question, because about a year ago, I moved all of our research to an earlier developmental epoch. So now all of my studies are now looking at people between 50 and 65 years old. And some of the work you're talking about is in people who are even younger than that, certainly. But at this point, we're doing a new pilot study with a ketogenic intervention in that age group, and we also have a blueberry study that we started in the fall, late summer, early fall, in that age group, and we have funding to do a strawberry study as well, and that's going to start probably next month, I think. And all of those studies are in this what I'm characterizing as a middle age group, 50 to 65. And I have two inclusion criteria. One is that they are overweight by BMI standards, so BMI of 25 or greater, and that they have subjective memory complaints, which means somewhat, we try to systematically measure this with some scales, but essentially it means a person would answer yes to the question, has your memory function declined over the last 2 years, 3 years, 5 years? Or so. In other words, have you experienced some age-related inefficiency in your memory capability? And then we exclude diabetics, but otherwise those are the inclusion criteria. And the reason to go back to that point is has a lot to do with what you mentioned, but even for people without genetic risk, it seems like health in midlife really predicts your health in late life, and that applies to risk for dementia as well. And so we want to catch people, 20 to 30 years before the onset of their clinical dementia.
Well, if you ever decide to do a trial in 30-somethings, happy to volunteer.
Okay. But I guess the implied answer to your question is yes. I think it would be extremely interesting to take a cohort of some of these people with family history or the genetic risk factors and see if a ketogenic intervention would work as well in those people as it has in our samples to date. I've looked at people with ApoE alleles, one or both alleles, in our ketogenic studies, just informally. I haven't published this, but I've just looked at those, and these have been small studies, so we don't have big subsets of people who are ApoE4 carriers, but I haven't seen a difference in their response to the intervention.
Interesting.
I know in some of the studies of Exona that medium chain triglyceride food, so to speak, they didn't get a nice response in the people who were ApoE4 carriers, but presumably those people were still consuming a standard Western diet and then taking the Exona in the context of that. And I wonder, I'd really like to do the study where you put all those people on a low carbohydrate diet and then use MCTs or Exona to augment it and to see if you can get a beneficial response, whether or not you're an ApoE4 carrier.
It's a perfect transition to the last question that I want to ask about ketones before we talk about your berry studies. But what do you think about MCT oil, exogenous ketones? A lot of people are talking about them today. The market seems to be flooded with MCT oil, exogenous ketones,
products,
derivative products, things like that.
Yeah, I think it's. It seems. It works. I think there's a biological basis for using it. The $64 question would be, though, what we just talked about a moment ago, which is how well does it work if you're still consuming some of the lower quality foods and the higher glycemic foods in your diet. I think a nice use of MCT oil is to potentiate ketosis in a low carb diet. But then the question would be, do you get as much benefit or any benefit at all, or how much benefit do you get if you're still consuming high glycemic foods?
Yeah, well, because it's like, once you actually show signs of memory loss, it's sort of like having multiple holes in the hull of a boat. And I think that MCT oil perhaps plugs one or two of those holes, but it doesn't have that broad biochemical impact that a ketogenic diet does in terms of reducing hyperinsulinemia, those epigenetic switches that we mentioned earlier about upregulating, or maybe it does. Maybe MCT oil does, because you're supplying ketones to the brain at a low level. We don't know, but
but the other thing about it that's perhaps fundamental is it's not attacking what we think of in some way as the root cause or one of the root issues, not all of them certainly, but which is this glucose hypometabolism because of an aberration of insulin and insulin receptor function. So that's nutritional ketosis does influence those parameters, which I
Interesting.
think, and that little bit of correlational data that I mentioned from our first study suggests that may be the case, that we did get that reduction in insulin, but it was also moderately correlated with their cognitive performance. So again, I've said this so many times today, but that needs to be investigated more. But it's a signal that maybe something's going on here, that we need to look at. The other thing I'll say is we're collecting data on this ketogenic diet approach in these middle-aged people, but we also completed a study as another pilot, small study, I think just 14 people with Parkinson's disease, early Parkinson's disease with mild cognitive impairment. And we didn't improve motor function. It doesn't appear that we've affected motor function, but we improved their working memory and their long-term memory function.
Wow.
And it's really a nice data set that I'm hoping to get to very soon to write up for publication, because I think it's another nice demonstration. So, I really want to stoke the fire in this field, because I think there needs to be more research.
Would absolutely agree with you on that. So if we can, let's talk about your berry studies. You're very passionate about berries, as am I, which is great. Why berries?
Well, actually, this goes back to another moment in my history, I guess. I read sometime in the early 2000s, which is getting to be a long time ago now, I read a paper by James Joseph from Tufts University, and his laboratory actually did most of the very early research, the basic science research on blueberries. And it was a paper that had been published in 1999. It was, I think the title of it was Spinach, Strawberries, and Blueberries Reverse Aging Changes in Rodents, or something to that effect. In the paper, he reported data in which he started supplementing different groups of animals with spinach, strawberries, and blueberries in late life. God, I don't remember how old they were at this point, but mice live 2 years basically, and these mice were about 1.5 years old, and he had a younger cohorts as well, and the supplemented animals performed as well on several cognitive and motor tasks as the younger animals.
Wow.
And that captured my imagination. It really really got me excited.
Can you comment on maybe what you think the mechanism is there? What is it about blueberries?
There's really quite a lot of basic science on berry supplementation, and especially blueberries, of course, but certainly others as well. And it seems to be the anthocyanins. Blueberries in particular contain 5 of the 6 major anthocyanins.
Interesting.
And so they're very rich in anthocyanins, and but anthocyanins are also in strawberries and grapes and blue corn and other things as well. Things with that deep purple blue color.
Yeah.
Often is a signal that it's anthocyanins, and they seem to have a lot of benefits for us with respect to the more well known is the antioxidant and anti-inflammatory benefits, but also they do some of the things we talked about with ketone metabolism. I actually have a slide in which I compare the benefits of both, and they're quite similar. And then one of my grander ideas is to do a comparative study of say blueberries or berry fruit supplementation against a ketogenic intervention. But in any case, they seem to improve glucose disposal independent of the level of insulin that the body produces, which is interesting. That's been investigated a lot in animals, but somewhat in humans.
Wow.
And they have the anti-inflammatory, antioxidant effects. There's one particular animal study that's very interesting. They looked at where in the brain the anthocyanins seem to go, the tissue that seemed to where they were distributed, and the hippocampus was one of those regions, which is a very important memory center in the brain, and the distribution in the hippocampus was actually associated statistically with the improvement in behavioral performance, cognitive performance. So this is a very powerful animal study demonstrating that that the anthocyanins in the brain, and they influence memory function, and they reside in the memory, those tissues that mediate memory function. So,
wow.
There's a lot of basic science work on mechanisms. We're just starting to accumulate some in humans. Actually, the studies, the NIH study and the second Highbush study that I mentioned, we were able to send urine samples to a colleague in Canada, in Nova Scotia, Willie Calt, who assayed the urines for anthocyanins, both the parent compounds, the food form anthocyanins, but also their metabolites. And there were up to 500 metabolites. Wow. Of the parent compound. So it's extraordinarily complicated. But the headline from this work is that the metabolic, one issue is that the metabolism of anthocyanins in the body after consumption goes on for days and probably weeks.
Wow.
And Willie thinks there's actually a shuttling between the bile in the gut and the liver of breaking down and then reconstituting these chemicals. And the body is just continually doing this for a long period of time, much longer than we had anticipated. The older data was, the older wisdom was that the anthocyanins were consumed and then they were gone in 6 hours. You couldn't measure them after 6 hours in the circulation. And that may be true, but then there's this extensive metabolic process. So the question becomes, is it the metabolic process? Is it what the organism is doing with the anthocyanins that's switching on the signals and producing the benefit? Or is it the acute consumption of the parent, the food form compounds? The data we have from these two studies, and one in particular, the Highbush in particular, suggests that it's the acute consumption, that it's the parent compounds that seem to be those, that the effects for cognitive benefit seem to be associated with that. But we can't rule out entirely a benefit from the metabolic process. So that's really exciting that we're actually starting in human studies now to be able to think about and get some data on the question you raised. Well, is it the anthocyanins? Well, yes, it seems to be from the basic science studies, but we're starting to see in humans now some evidence to support that as well.
Do you have any data on berry types, varieties that have the highest concentration of anthocyanins? Is it wild blueberries? Does being organic or versus conventionally produced matter?
I'm not sure that there's been a demonstrated difference between wild and cultivated blueberries, but Jim Joseph actually told me the story once. He was working with Ron Pryor at the Tufts lab, the USDA lab at Tufts, and Jim was looking for a new research line to get into. This was before he was doing the blueberry work, and Ron had developed the ORAC index, which is that index to measure antioxidant capacity of food substances. And they used to commute to work and back home.
Wow.
And together. And so Jim said he asked Ron which berry that he had looked at had the highest antioxidant index, and Ron told him it was blueberry. So he decided to look at blueberries based on that. Now, I've heard people say that acai berries and other berries have potentially higher antioxidant capability than blueberries. I'm not sure that matters so much.
Hmm.
My point of view is that berries are healthy and they have benefits for us, and that it's a good idea to consume berries on a semi-regular basis to get some of those benefits. And our data in terms of cognition with aging suggests that it may provide enough benefit to mitigate risk for late life dementia. Um,
That's
Again, not
incredible.
proven, but there's an accumulating evidence for that now.
Amazing. Well, my last question for you then would be, knowing what you know about how important diet, and these things like blueberries and MCT oil and all that stuff is for the brain. What is your diet like? I mean, what's like a day in the life of Robert Krikorian look like in terms of the kinds of things that you eat?
I think there are two principles to a healthy diet. One is you have to eat vegetables. Vegetables are crucially important because they have so many micronutrients, vitamins and minerals, and they have a lot of fiber, which is essential. I mean, a lot, the most taken medicine in the world, I think, is Prilosec, you
Really?
know, which inhibits the gastric juices to break down fiber. I mean,
Oh my God.
it's so beautifully ironic, um, that's a whole other hour talk, I guess. But I mean, there's a lot of politics in food and in Western food production and the application of technology to food, which I think um, is in a very important issue. But in any case, vegetables are crucially important for a healthy diet. And the other thing that goes hand in hand with that is to avoid processed foods as much as possible.
Hmm.
Um, but what I do is I eat a lower carbohydrate diet all the time. I generally eat 2 meals a day, not always, but most of the time it's 2 meals a day. So one meal mid to late morning and the other meal early evening. And I try to squeeze in those 2 meals when I can, within an 8-hour period. So I have a 16-hour fast of no calories. Um, and I eat, I eat protein. I eat meat. I don't think meat is terrible. Um, I don't worry about fat, except I avoid trans fats and most polyunsaturated fats like corn oil, etc., and soybean oil, those kinds of things. I eat as much fat as I can. Is actually the biggest problem is my diet is not eating enough fat. I mean, I eat a half to a whole avocado a day and try to use olive oil as much as possible. I do use coconut oil a little bit, but probably not. I probably could use it more if I thought about it more. Um, but I eat vegetables once or twice, usually twice a day, but often, but occasionally once a day. And I focus on kale and celery and different kinds of lettuce, and then throw in peppers and other things, broccoli and so forth, as much as possible. But the fundamentals are kale and celery, actually, which is
Hmm.
kind of interesting. And that's just where I am now, and that could
Uh-huh.
change. And then I eat a lot of nuts and fish. I don't take an omega-3 supplement, but I eat sardines and salmon 2 to 4 times a week.
Hmm.
Um, and I don't take a lot of supplements except vitamin D3 because I'm in an office all the time, and magnesium because it's hard to get magnesium even if you're eating vegetables, because I guess the levels in the vegetables are reduced now to relative to what they used to be.
Interesting.
And magnesium is very important, especially for ATP production and so forth.
Hmm.
Um, so that's it. And of course, I don't take any calories from fluids, juices, or things like that. I don't eat any desserts. I don't eat any grain products at
No
all.
grain products, huh?
No, zero.
I'm the same. But I think it's funny because there's still, when you read about things like the Mind diet and other forms of research, they tend to
Mhm.
advocate for eating
Right.
lots of
They
grains.
advocate legumes and whole grains, and I don't eat any legumes either.
Hmm.
Um, I think I think they're not a great bet calorie for calorie in terms of nutrition. I think it's hard to beat kale
Yeah.
with soybeans or other beans or or whole grains even. And to some extent, whole grain is a marketing concept. I think. I mean, it's on television, so I kind of don't trust it. Um,
I love that.
but and then the other thing I do is 2 times a year I do a ketogenic diet for about 8 weeks. So I drop out all fruit when I do that.
Wow. In general, do you eat other sweet fruits aside from blueberries and
I eat
strawberries?
berries. I eat apples, but I just eat the apple peel, and I give the rest of the apple to my dog.
Wow.
Um, Occasionally, not that frequently, I'll eat an orange, but that seems incredibly sweet to me, actually.
Yeah.
But there are benefits for those things. I mean, the when I'm not doing the ketogenic phase, I will allow myself some of the other fruits.
Hmm.
I love plums and peaches, and those summer fruits are great. So,
Yeah.
um, and I think there is benefit, and they're actually relatively low calorie. When you look up the total calories in a plum, it's like 45 calories. It's not terrible.
Right.
Um, so
Very interesting.
I do.
Cool. Well, we're very similar in our dietary approaches.
Yeah.
So, I love it. I don't think I have any other questions. So, I mean, unless there's anything that you want to add that you feel like you haven't gotten to talk about, but this was fascinating.
Yeah. No, I guess that's good enough. I mean, I think we could, if you asked me another question, I'd talk for another 30 minutes. So
Yeah, we could probably
maybe
go
we
on for
should
hours and hours and
we
hours.
shouldn't do that. Yeah, maybe save it for another time.
Exactly.
Yeah.
Exactly.
It
Cool.
was really nice to see you and talk with you.
Same. I loved this. This was amazing. And I really appreciate your time on this beautiful Sunday when I'm sure you could have been out, doing other things. But thank you.
You're welcome. Welcome. I'll be happy to do it.